monkey is there. Fish can run. They are only playing. There are some monkeys. They are playing continuously games. I'm victim as power. My laptop does not stays with me. Problem is this. Who told you that? This power information who gave you this? प्रिंस महाराज की जय। आप कहाँ पे थे? गुड इवनिंग प्रिंस। अब ये मत पूछना आप कहाँ थे क्या थे। Good evening, everyone. Come and join the session fast. Now listen. Yes. Hey, understand this. When we are discussing about when we are discussing about see, if the people will understand, we discussed homozygous and heterozygous. We learned that homozygous and heterozygous two are there, correct? Na? So also we have exactly had a discussion like when we will be talking about homozygous, okay, when we'll be discussing about homozygous and heterozygous, okay. So now next part we are going to understand like this already we have learned discussed like yes, characteristics we talked about which type of traits are dominant and which type of, yeah, that will be blurred now because it is taking time to download. So which type of traits are homozygous, which are exactly, so we know that dominant traits will be there. Yes, Ashwita, good evening beta. Govika, long back I have not seen you beta. So now look here. So we are going to talk about dominant and recessive traits. We came to know that some traits are exactly dominant and some traits are recessive traits. Okay. So recessive traits are suppressed traits. Suppressed traits means, hi Govika. So suppressed traits means those traits which are not received if they are single. Understood? So they must be in pair, then only they will be what you call, they will be they must be in pair, then only they will be selected. I hope you might have understood this one. So now we are going to talk about the next part, like homologous chromosomes. You see the chromosome stands. Yes, Ashwita, please come on live. So, homologous chromosomes already I discussed. This is centromere, and this is exactly the central part is called as centromere, and this part is called as telomere. And you will be seeing a drastic similarity between them. So, such type of traits will be exactly called as homologous chromosomes, where the traits or the characteristics are more or less similar. Okay, Satya, good evening, beta. So these are chromosome pairs for approximately same length. Centromere positions are staining pattern. Can you see the staining pattern? The staining patterns are all same. Okay, Sophia, good evening. After that, with genes for the same characteristic as corresponding loci. Can you see the corresponding loci, the locations, the things are exactly looking more or less same. Now, in an organism, one homologous chromosome is inherited and for mother and one from father. So definitely the traits should be exactly coming. So now last class also we have discussed that part. So I'm quickly shifting to the other sections. So when other sections I'm shifting, so what and all are the next point we are going to discuss. So genome. So if somebody will be asking about genome, the total number of genes present in the haploid set of chromosomes, that is chromosome gamete, it is exactly considered as genome. Okay, so what is beta? The total number of gene present in the haploid set. How many gene will be present? Like this, first we have seen the strand. Now how many gene will be present on the structure, that will be considered as what? Haploid set. What it is considered as? Haploid set. Okay, so now we will go for the next part. Okay. Yes. So now beta progeny. Progeny means if somebody will be asking you, good evening, only I'm seeing five people live. I don't know how many children are live right now. I want to know the names of all the people. Progeny means offspring. Yes. Progeny means offspring. Offsprings means the cookie babies are exactly considered as what beta? Progenies. Okay. Now what are factors? So factors are the characteristics. You need to understand this one. If somebody will be asking you about factors, so these are the gene located on the chromosomes. So the gene located on the chromosomes are considered as factor. The gene located on the chromosomes are considered as factor. Okay. So now we are going to talk about locus. Locus means the location of a particular gene is considered as locus. Understood? Next is alleles. So it represents two alternate form of unit factor. Two alternate form of what? Unit factor. So they will be considered as alleles. Now, but here it represents two alternate form of unit vector. And you know now, what and all are the alternate form? Can anybody tell me how many type of alleles are there? Dominant and recessive. So if somebody will be asking you how many type of alleles are there, so we are going to say dominant and recessive. How many type of alleles are there? Dominant and recessive. So dominant and recessive are the two type of alleles. Now homozygous and homozygote. Homozygous and homozygote. So if somebody will ask you homozygous and homozygote, an individual with identical alleles at one or more loci or homologous chromosomes, it will be called as homologous or homozygote. This is most important beta. So you need to understand one thing. Okay, an individual with identical alleles. So both either has to be T, capital T, or both either has to be small t. So this is exactly considered as what? Homologous or homozygous. I hope you might have understood beta this one. You scared me. Okay. Yes. So homologous or homozygous. So either both capital T or both small t will be considered as homologous or homozygous. So this part we have discussed. So now heterozygous, one capital, one small. So either one capital will be there or one small will be there. It will be considered as homozygous or heterozygous or heterozygote. Heterozygote, it is considered, yes, capital T. So values constitute of genotype are different. Identical means homo, different means hetero. Identical means homo and different means hetero. So now dominant trait means these type of traits are that type of traits, they can exist single. Understood? So they are called as what? Dominant trait. And recessive traits are those traits which are expressed as what you call, if they have to be exactly always in pair, so they are always be exactly present in pair. So this part you need to understand. Now recessive alleles, they can be expressed single, they need to be in pair. Understood? So what they have written about it is represented by lower caslets. Caslets means in lower letters, like alphabets we are using now. Capital letter is for dominant, small letter is for recessive. So dominant can exist single, but recessive need pair. Recessive need what? Pair. So now we are going to talk about genotype and phenotype. See, what is the meaning of genotype? If somebody will ask you, yes, dominant can exist single. Dominant can exist single. So, what is the difference between genotype and phenotype? If somebody will be asking you genotype, okay. So now, beta, what is that meaning? So now look at it. When somebody will be asking you genotype and phenotype, so we are going to answer this genotype and genetic constraints will be there, that will be considered as genotype, and the physical part will be there which is visible. It is called as phenotype. Like for you, I can say that sometimes your parents are exactly looking. You are looking like your parents. So your relatives are telling, hey, you look like your father, or your nose look like your father, your eyes look like your mother. Correct, na Porani? So many times people are telling you are looking like your grandfather. That day I saw your grandfather and I understood that Porani looks like exactly like grandfather. Correct, na? Huh? Huh? His grandfather, he knows Hindi. But only one thing he did not look from his grandfather. Grandfather height is so good. Okay, no problem. Those people, science exams only tomorrow. Today, science I am going to do. Tomorrow morning, physics, electricity. Okay? Yes, tomorrow morning, physics, electricity is there. Don't miss that class. Listen nicely. Tomorrow morning, physics, electricity is there. Tomorrow morning, physics, electricity is there. Now, look at it. Phenotype and genotype done. Now, F1 generation and F2 generation. F1 generation is called as first filial generation, and F2 generation is called as second filial generation. F1 is first filial generation, and F2, okay, which chapter is there? You inform me, I will give revision of that. So F1 is considered as first filial generation, and F2 is considered as second filial generation. Understood? So now there is a progeny resulting from a crossbreed. So this is exactly the first generation, and second one is the second generation. Now we are going to talk about Mr. Mendel, who is called as not Mendel. It's Mendel. Mendel is considered as the father of genetics. Understood? Mendel is considered as what? Father of genetics. What is yeah? Light chapter and human and colorful world. Let's have a fun with tomorrow's session. Father of genetics. Yes. So father of genetics. Yes, the genotype is inherited from the parents. The phenotype is not inherited. AI. Phenotype is coming because of genotype. Who told you? Kritika. Phenotype is because of genotype. I look like my parents because of my genes. Genes are not visible, but phenotype is visible. That's the only difference. You people have to understand that. Don't write wrong in examination. Here people will approve your answers, there people will not approve your answers. This is a very clear cut answer I'm giving you. Genotype is exactly making phenotype. So phenotype depends upon what? Genotype. How the gene will be there, how the DNA will be there. You will be looking like that only, na? So how can we say that if phenotype is independent? Yes, phenotype is not independent. Phenotype is dependent on, yes, phenotype is dependent on genotype. So now we are going to understand that Mendel is known as father of genetics. He was the first scientist who has made a symmetric study of pattern of inheritance of characteristics from parents to progeny. Means what type of characteristics or what type of traits are transmitted from parents to offspring was exactly told by whom? Mendel. So Mendel was the first one. So now his birth time and all this are exactly this PDF, and it will be reaching you. You will be getting these points. So now overall chapter revision means one thing we are doing. That thing you will come to know very soon. Okay. So now look at. So in 1843 he was admitted to that is Augustinian monastery of Saint Thomas in Brunn, taking the name of Gregor. So Gregor John Mendel. John Mendel. So he studied in the University of Vienna, where he studied natural history and mathematics. Mathematician. Courage. Understood. So he experimented, and he is exactly selected. He has selected tree plant. See this now. Wow, mathematics. Underline. Is the network okay? the best part is this one, yeah. This one. He studied history and mathematics. This is the best part. Okay. This is the best part. He studied history and mathematics. Mathematician. He was mathematician. So only he did experiment. Understood, no? Okay, fine. Let's go for it. Hey. So he performed his first, okay. Now research phase lasted till 1868. After that he got kidney disorder and he died. Pavam. So now Gregor John Mendel had a great interest in hybridization of plant. Yes, he noticed that certain traits in garden pea plant. What is the scientific name of garden pea plant? Payasum sativum. Sativum. So Payasum sativum. So you know, like Tamil, what we call. So in Tamil you can sow what do you call Payasam saptam. Yeah, this actually some of your senior Annas and Akkas, they told if I want to remember means I will remember like this Paisam Saptam means Paisam Satyam. Understood or not? So only I am telling. So this is exactly what Paisam Satyam. In the garden of church, he performed certain experiment of hybridization of about how many years? 7 years. So he maintained a proper record of his result and then made certain generalization and put forward certain laws of inheritance in 1866. So certain laws of inheritance he put forward for 1866. Okay. So Jotham, my first question is, what is the scientific name of pea plant? We'll see Jotham is present in online or not. What is the present? What is the name? Payasam sativum, not Payasam saptam. Sativum, Payasam sativum. The name is Payasam sativum. Yes, the Payasam sativum. Yes, Payasam sativum. Absolute. The correct answer is Payasam saptam sativum. Now look here, beta. Okay, the question what they are asking again and again, enough. The question what they are asking again and again, why he has selected pea plant for his experiment. He could have selected any other thing, but why his selection was for pea plant? Very good. So we will only underline the most important point and point wise we will write in science exam. The first point is garden pea plant distinct, easily detectable contrasting character. This should be your first point. Distinct means different. Easily detectable means easily catched. Means easily catched. I am saying easily seen. Understood? And contrasting means different character. So this should be your first point. Short lifespan, don't go for that. The most important point, see now onwards, whenever you are writing science exam, you will be writing first point at first, second point at second. First is distinct, easily detectable contrasting character. Understood? Easily detectable means you can be able to catch it out. The plant reproduces well and grow maturity in a single season. This is the most important part. Gaining maturity in how many seasons? One single season. The plant reproduces well and grow to maturity in a single season. First is the detectable contrasting character. Second, one single season, it is over. Third, it is self-pollinating. Understood? It is what? Self-pollinating. It is what? Self-pollinating. Self-pollination could be prevented by removing the male reproductive part of the flower. So how can we prevent it? By removing the or preventing by removing the male reproductive part of the flower. So if we will remove the male reproductive part of the flower, the self-reproduction will be stopped. I hope you might have understood this one. Is it done? Very good. Yes. Now we will go for the next one. Cross pollination could be done artificially. Cross pollination could be done artificially. But self pollination, how we can prevent? By remove the reproductive. First point, We will write point wise. The first point is garden P has distinct, easily detectable contrasting character. Got it? Second point is the plant reproduce well and grow to maturity in a single season. Third, the pea plant is self-pollinating in nature. What nature? Self-pollinating in nature. Self-pollination could be prevented by removing male reproductive part. Systematically right. Next, cross-pollination could be done artificially. So cross-pollination could be done artificially because the size of the flower is small and hence it is very convenient. Next, Mendel took different varieties of this plant showing 7 pair of contrasting characters. The characters selected by Mendel for his experiment are of following table. So now we will see the character. Did you understood? First part, everybody might have understood. First part is distinct, easily detectable, contrasting character. Yes. Second is single season they are needing. Understood? Third is self-pollinating. Fourth is we can remove male reproductive part. For cross-pollination, artificial technique can be utilized. Then different variety of pea plant, what he has taken, it will be having different contrasting character. So different characters, contrasting character. Yes, thank you for liking the class children, but I am seeing only 7 children now online. Come on, check this one out. We will see the 7 contrasting characters. Come on, catch this. This PDF will be available to you, so you need not to be worried about. This PDF will go to you, reach to you. Okay? We people are making a separate all the 10 standard student group. Final revision, all the 10 standard. Only one link will be provided. Videos will be private. Anything we are changing. Okay? This week itself, Sunday classes will start. Huh? Yeah. No holiday. Huh? Sunday, morning, afternoon, both time. Because we have to finish humanities on Sunday itself. Okay, so we planned like that only. Color of the pot, now look here, beta. Because electricity, carbonates compound, this week 2 topics we are going to finish only. Topic finishing now we are doing. This part once finished, this chapter will be over, correct now? After this, we need to go to carbonates compound and finish that part. After that, electricity and magnetism. That's it. Got it? And we will start revision parallel. Maths revision we will start. Because sector and segment we have already studied. Yes, the first is form. Grey and brown, wrinkled and white and wrinkled. You might see some pea plants are white and wrinkled. Wrinkled means outside you will see some like baby line. That is called as wrinkle. Next one is cotyledons will be there, yellow color or green color. Next flower color will vary, white color or violet color. So these are the character. Next one is forms can be seen. This is the pot shape. Pot shape can be either like this or pot shape can be constricted. Pot shape can be yellow color or pot shape can be green color. Pot shape can be full or pot shape can be constricted. Stem, axial pod flower along and here terminal pod flower tops. So you can see axial is on this part. Nodal, between nodes and internode region, you can be able to find it. Or here you will see the terminal, that is the upper sections. You will see the flower. So this is difference in location. In simple term, what we can say? Difference in location, and second is the size, height. Understood? The height of the plant also count a lot. The height of the plant is also something which is counting a lot. I hope you might have understood this one. Come on. Till here everything is clear. So these are the 7 contrasting characters. First form, or the right round up 2 colors. What are 2 colors? Color of flower. Forms, spot forms, fuller and color and yellow and green. Then either in axial location or terminal location. Then size either short or long. So either short or long. Understood. So these are some things which we need to learn. Next we will go for Grigor John Mendel. It will take 10 minutes. So why Mendel selected pea plant for his experiments? Answer is reason. In addition to his choice of suitable part, he took a number of precautions while performing his experiment. He restricted his experiment to one or very few pair of contrasting traits. at a time. See, where people will try to merge different contrasting character at time, what Mendel did is he took one character at a time. So that was acting as a benefit. Why it was acting as a benefit? Because one trait taken at a time means the risk of failure or success is very high. Correct now? So failure part will be less now, because if we might have merged more characteristics together, that time the chances of failure might be very high. Understood? Failure might be high that time. That time the chances of failure might be very high. But now the chances of failure is not so high because single character he has selected. So now we are going to talk about Gregor John Mendel, father of genetics. Now selection of pure parent. First part what he did. Understood? So pure alleles first he selected. First he selected what? Pure alleles. Pure means like how many of you have seen Harry Potter? In Harry Potter, you might be hearing that one Mughal blood will be there, pure blood will be there. So mixed blood. Mixed blood means recessive traits. Hey, mixed blood will be recessive traits. Hey, just telling you so that you can understand. In simple word, means either both dominant or both recessive. They will always be called as homozygous. What they will be always called as homozygous. They will always be called as homozygous. So now look here. So hybridization of parent plant to produce F1 generation is done. So now look here, beta. Raising of second and subsequent generation by self-fertilization. This is first one is effort. First one is done with effort. Second one is obvious. Understood? So now we will go for the next one. So this is called as monohybrid cross. Everyone, please go for monohybrid cross. Monohybrid cross. Understood? So okay, monohybrid cross means one single part. So cross between two individuals which differ in only one pair of contrasting character is called as monohybrid. So either we will cross for height, tallness or darkness, or we will cross for color, yellow pod or we will cross for what you called understood, or wrinkled and round means different type, seven characteristics within a axial and terminal. So all these characteristics were there. So I hope you might have understood this part. So this is exactly some things which you need to understand. I hope till here you might have understood this part. Is it done? I hope you might have done till here. So formation of only one character, one at a so actually this is one. So hybridized inheritance of only one pair of contrasting character. So he selected pure line. That's what I told you now. Homozygous one time. And pure line and put them into cross pollinated such plant having contrasting trait, considering one character at a time. For example, Mendel crossed a pure breeding tall plant with a pure breeding dwarf plant. So pure line has to be selected. So he selected pure line. So Mendel crossed pure breeding tall plant and pure breeding dwarf. So F1 generation is exactly totally understood. So it is exactly totally pure line. I hope you might have understood. Now we will go for the next one. Okay, slides over. So this is part 2 done. We'll go for the next one. Only monthly stands celebrate like this. I can't understand this video. All one side, monkey one side. It's coming, coming, coming. Wait. So let's go for the first one, Mendel's experiment, monohybrid cross. First we will go for monohybrid cross. In monohybrid cross, please kindly check how the cross is done. This PDF will be available to you. All the PDFs will be available to you. Your one common group will be found, and in that common group, only those students will be there who is continuously attending the classes. So I'm making a final group. In that final group, the shortlisted selected students will be for the final 90 days game. Okay. So now it is exactly done, and we are planning for that. From tomorrow onwards, you will be seeing the changes. Got it. So the final group will be where only PDFs will be sent, materials will be sent chapter wise for final preparation. Are you all ready for that? And once we will announce one chapter you have to prepare means you have to prepare that chapter and then revision will start with that. Got it? So first we are going to finish this chapter, then with revision we will start. Hello sir. Huh? One of my friend is there. He is very finest photographer from Germany. Manate. His photographs one time I will show you. He is online. Yes, yes. Some of his creative. He is a very creative person. Okay, I'll send the creativity of his in my status. Manak, send some photos taken by you. Some of the people wants to see that. Now look at so Mendel pea plant. Now this is Mendel's pea plant experiment in the F1 generation. If you see, one by one we will do that. This is phenotype. Phenotype is physical appearance. Physical appearance here means tall and dark, but it is a pure line. Pure line means both the trait has to be either dominant or both the trait has to be either recessive. So quickly just check this one out. So here genotype is TT or small tt. Got it? So now next one is gamete formation. Gamete formation at the time of gamete formation, this is phenotype, genotype. At the time of gamete formation, you can't take the pair. You have to take only one. Understood or not? So here at the time of gamete, the first filial generation, only one T and T is taken. Got it? Now, this T will give you only TT. Means all tall will. Understood? But are they all pure? No. They all are mixed. They all are mixed. So now, next time, so this is monohybrid cross here. What we are doing is, we are taking the characteristics and now we are cross breeding it. Understood? Now we are doing what? We are cross breeding it. So the monohybrid cross between tall and dwarf pea plant, the symbol TT designed like dwarf and tall, and all individuals are shown in the rectangular part. So this is exactly done. Now look at it. So this is capital T small t. Now second self cross. First we got this one, same what we got in the first generation, F1 generation, all will be tall. In F1 generation, all will be tall. But now we are going to go for F2 generation. In F2 generation, again same mixed will be crossbreeded. This time, this TT will come as capital T. Can you see? So now self. So this is exactly single TT taken now. So one single T and T will come. One single T. Now this T and T will merge. Understood? So this is tall. This is pure. This is pure, and this is mixed, and this is mixed. But here, how much tall is coming? 1, 2, 3 tall. So ratio of tall versus short will come as 3:1. Understood? Pure versus mixed. 1:1. Pure versus mixed is 1:1. Pure versus mixed is 1 is to 1. So this chart I will ask you at the time of conducting test. Understood? So this PDF will be available to you at the time of conducting test. I will be asking you this one. Okay? I hope you might have understood. So now this is exactly called of observation. So based on the experiment of monohybrid cross, what is all observation he has done? So wait. Something I missed. This is the first part. After first one, some lines were there. This slide I should do. Okay. So a cross between two individuals differ only one pair of contrasting character is called a monohybrid cross. So monohybrid cross will be exactly done where one pair of contrasting character is seen. I yes. But first you come today to gravities in class. Wait, I'm sending an email. So now look. So when somebody will be asking you this contrasting character, just 1 second, but I need to transmit this information. It is very important. Okay, but now listen. Yes, so we are checking one contrast. When mental cross homozygous tall plant, we saw that first generation is all tall. But are they pure or mixed? Mixed. After that, when he went for second generation, he tall versus darkness is 3 is to 1. That is 70 25%. In monohybrid cross, for different contrasting character, the result were same. Now we are going to check this part out. So that is now after that, now Punnett square. See, Mendel, based on Mendel's observation, one Punnett square has been made. So that is Reginald C. Punnett devised his approach and he has made one square rule. Can you see the square rule? Plate part, how he has made in a very simple contrasting part. So this is rule is called as Punnett square rule. Punnett square rule is there in NCERT book?
Yes, ma'am. Checkerboard.
Yes, the checkerboard. The checkerboard part is exactly considered as Punnett square. So here this Punnett square part we will be checking and we will be writing. Okay, done. So now what we will do is Punnett square. So Punnett square or checker diagram in which the different type of gametes of one parent are placed along one side of the square and those of another parent are placed along the other side of the square. The possibilities will be exactly genotype, gamete or subsquares. Understood or not? So we will go for the next part better. So after that, now with this Punnett square rule, now, it is the same like but it is exactly made in a box pattern. This TT will come TT, this TNT will come TT, this TNT will come TT and this TNT will come TT. So that's the color, the checkers board. Up and down. That cross is there, that's maybe. Refractive index questions. Okay. So, but a Punnett square rule is exactly done. We talked about Punnett square. Next part we are going to do. See, the Mendel has discovered three important laws he has made. So, what are now laws? Let's talk about the laws. We discussed very important law. First law, what we talked about is we derived the following three postulates of principle of inheritance by using result of monohybrid clause. Okay. So the three important laws are principle of paired factor. What is the first law? Principle of paired factor. The first law is called as principle of paired factor. The second law is called as dominance, and the third law is called as principle of. So what are the three laws? The three laws are principle of paired factors, principle of dominance, principle or law of segregation. Law of segregation. Okay. The first person who will be added in the group, new group, where final examination preparation will be done, is Ovian from Anupuram group and Kavya from online. They are very attentive children. Disturb me as much as possible. Yes, only one group because most of the PDFs and everything has to be given. Okay. Are you all ready? With our first law, we are going to understand principle of paired factor. The alleles are always exist in pair. They can't exist single. So that's why it is called as principle of paired factors. So genetic character in an individual are controlled by pair of unit factors or genes, for which it is inherited from and other from the male parent. So for the two factor controlling one character may be similar or dissimilar. That's what similar means. When we talk about similarity means what it is. Either both dominant or both recessive. So here this part is very important. Controlling one character may be similar. That important factor is there. So this is exactly coming for genetic factors. Got it? Now we will be going for principle of paired factor. Paired means always first only we have discussed. So it will be paired factor. Second one is principle of dominance. Who can't dominate who? So like dominant trait is there and recessive trait is there. See either both are there till dominant. This is also dominant. Here also, if all dwarf is there, then two dominant. But if this is exactly small, then only it is short. Otherwise, all T, single T can, capital T can dominate all one. I hope you might have understood. This is called as dominant trait. This is called as what it is? Dominant trait. So now then one allele is dominant over the other. Now next one, when he has taken law of segregation. So what is the meaning of law of segregation? You need to understand this law is very important. Type about, note about law of segregation. The paired alleles of a unit factor separate or segregate randomly during gamete formation. So each gamete can either receive either one or two alleles of a unit factor controlling one character. So definitely beta, this is the most important part, note down. This is called as law of segregation. The paired alleles of a unit factor separate or segregate. Understood? So pair will what? Separate. Did you saw na separation? TT was there. Then how much would T we have taken? Only one. So first it is separated. Correct, Vishwa? It is separated. After separating, each gamete can receive either of the two alleles. Each gamete is receiving how much? Either of the two alleles of unit factor controlling one character. So here it is exactly seen. You see, it is separated, and after separated again it is exactly showing one character by joining together. So this is called as what? Law of segregation. What this law is called as beta? Law of segregation. Absolutely correct, both of you. Yes, beta. This is called as law of segregation. So this section is called as law of segregation. Now, when two contrasting characteristics are taken, can you see here when two characteristics has been taken. One characteristic is round, another one is wrinkled, and then yellow color and green color. So these are exactly the two important contrasting characters. When you have taken two characters, how many are exactly present? Second generation. First failure generation, cross pollination, one part is opted. Okay. When second generation, second self pollination happened, then in the Punnett square, how many are coming? Total check it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 13, 14, 15, and 16. So total how much is coming with a 16 it is coming. Understood? It is coming how much? So parents were cross-pollinated. F1 generation was raised in which all the plant produced had round and yellow seeds. Round and yellow. First generation was round and yellow. But second generation, can you see yellow round 9 out of 16. Green round 3 out of. Can you see? 9 out of 16 is and round. Green round 3 out of 16. Yellow wrinkled, green wrinkled. So green wrinkled amount is very less. Okay. So this is exactly the Punnett square constraint, and we have understood this is F2 generation. This is what generation, beta? F2 generation. So now they talked about F2 generation. Now just check this one out. What we have studied. So by self-pollinating F1, F2 was raised, we found 4 combinations. How many combinations? 4 combinations. Before monohybrid, how many combinations is there? 2. Either tall or tall. But when he is crossing two characteristics together, it means how many contrasting characters came? 4 contrasting characters. Yellow wrinkled, round say, yellow wrinkled, green round, green wrinkled. So now, based on this now, he has postulated the fourth, he has postulated the fourth law. It is called the independent assortment. That's a very important one. This law is called as law of independent assortment. What it is called as law of independent assortment. This PDF will be available to you children. Okay. So what is the meaning of law of independent assortment? This law states that the allele of two different characters segregate or assort independently. Understood? It will be independently segregated. Then after segregation, they will independently get chosen. Got it? So this is called as law of. This law states that any pair of unit factor of gene controlling different character can segregate or separate independently. You don't forcefully do it. It will itself segregate and will itself choose. So that's why it is called as law of independent assortment. So one pair which unit factor is received, it will itself be chosen and it will itself be reflected. Like round and wrinkled, yellow and green. So independently yellow and green is coming, yellow and round is coming, yellow and wrinkled is coming. Correct? Na? So this is called as law of independent assortment. First generation it was all yellow and round, but second generation it became 4 contrasting characters. Second generation independently it selected. Independently selected. How many characters? 4 characters. So what name it has been given? Law of independent assortment. Now, how did this trait get expressed? But there must be something to express the traits now, because suddenly we cannot express the traits. Understood? So this traits has to be expressed. So the reason behind is cellular DNA. What is the reason? Cellular DNA. So cellular DNA is the information source for making protein in the cell. This is the most important part, children. We are in a very critical section. So you must remember this. What is the main thing responsible for expression of a trait? The main thing responsible for expression of a trait. The main thing responsible for expression of a trait is this only. I hope you might have understood. Understood. The main thing responsible for expression of a trait is what, beta? Cellular DNA. What it is called as cellular DNA. One more. Cellular DNA. So cellular DNA is the formation source of making protein in the cell because information has to be transmitted now. Information has to be passed on. Now a section of DNA, deoxyribonucleic acid, that provides information for a particular protein is called as gene for that protein. Now, character of organisms are controlled by protein. Height of plant depend upon the amount of particular plant hormones. Are you understanding? So, first is the gene and the second is the hormone. The second part is the hormone. The amount of plant hormone produced depend on efficiency of enzyme that is involved in this production. So, efficiency of enzyme is one of the most important thing. And this enzyme, so the second, this 2.9 should be there in your notebook because these 2 points and point-wise representation you should be writing. Like cellular DNA is formation source of making protein, and the DNA section of DNA will provide this information from one gene to the another. Second is the organisms, the characteristics of any organism is controlled by whom? Protein. Protein is exactly controlling the characteristics. Understood? Like height of the plant depends upon the amount of particular plant hormone. Now, amount of plant hormone produced depends on efficiency of enzyme that is involved in the production. Next, if the enzyme works properly, required amount of hormone is produced and the plant will be normal height. But if the enzyme is not working properly, it will not be proper. I hope you might have understood this one. So now, how do this trait get expressed? Third point is, if the gene of that enzyme has an alteration, that means over, Govinda Govinda. Understood? A little alteration will be there, means that makes the enzyme less efficient. The enzyme is not that much efficient. So the amount of hormone will be less and plant will be short. Good amount of hormone means plant will be tall. Less amount of hormone means plant will become short. So that's the most important thing. Thus, gene control characteristics of trait. So characteristics of traits are controlled by whom? Gene. Next, contribution of male and female parent in expression of characteristics. So many times they are asking this question that what is the contribution of male and female parent in contribution of or giving of characteristics. The answer is considering the rule of inheritance, both the parent contribute equal amount of genetic material. Understood? So how much equal amount of genetic material is transmitted from parent to the offspring. Okay, so such trait is influenced by both maternal and paternal DNA, means from mother side and father side. Each cell will have two copies of acrosomes. Note down. This is called as homologous chromosomes. That is called as acrosomes. Coming from the father side and mother side. Tomorrow morning, what subject you people want to study? Inform me. Okay. Maths are even start. The science 4 chapters are little bit left. We need to finish this one. Okay. Now look at it. Each cell will have 2 copies of chromosomes. So haploid means and diploid means 2n. Haploid means one single. Diploid means pair. So pair is always taken. So for DNA, pair is exactly taken. The mechanism by which sex of an individual is determined is called as sex determination. What is called as sex determination. So it will be determined what? Genetically. So chromosome inherited by male parent determines whether offspring will be boy or a girl. So it is totally determined by the male, not by the female. So Mendel has exactly expressed. So sex chromosomes, it will come as autosomes. Autosomes are the other factor. 20 total 23 pair of characteristics are there. So 22 pairs are called as autosomes. 23rd pair is called as sex chromosomes. 23rd pair is called as sex chromosomes. Can we see here? So here when you see sex chromosome will be exactly either allosome or idiosome or heterosome. So can you see X for female always same, but for male will be short. Understood? So whose chromosome is exactly determining the sex of a child? Male. Because see, Y will be only present in male. For female, always X will be there. When XX will come, it will be human female. When XY will become, it will be human male. Understood? So female can give X single. Male can give either X or Y. So the chromosomes are divided into 2 parts. What are they? X and Y. So male, what you call chromosome is of 2 types, 50% X and 50% Y. But for female, 100% is X. So 50% chances of a female child or 50% chances of a male child. So it will be exactly characteristics is that type. Now we will go for the next one, autosomes. Autosomes means what? So chromosomes other than sex chromosomes are called as autosomes. So how many autosomes will be there? 22 pairs. 23rd pair is sex chromosome. 23rd pair is what? Sex chromosome. That so this mechanism of sex determination in human being is called as Ligas type or XX female, XY male type. So this is exactly, and it is total 100% is not there. It is 50%. Can you see that 44 characteristics are there? 22 plus X, 22 plus Y. So here it will be 22 plus X. So definitely boys chance and having and girls chance having is 50-50. Got it? It's not like so female are not the contributor of having a boy or a girl. It's about the male. I hope this is the part. Only blood group one time I need to check. I'll be informing you blood group. So this is the end of this chapter. I hope everybody enjoyed this class. This PDF will be once we will shortlist the final students. This PDF will be available. Okay.